Okay, so girls, that day we had already studied how a peptide bond is formed between the two amino acids, okay, between the two amino acid residues. Today we will study the structure of the peptide bond, okay. We will study the structure of the peptide bond. So as you can see here, there are three amino acids. You can see this is the first one, this is the second amino acid, and this is the third. So we will take a look at we will take the example of a tripeptide. Now in this tripeptide amino acid, as you can see here, we have to remember the geometry of the peptide bond. It shows that the carbonyl oxygen and the amide nitrogen are trans to each other. Okay, this is the carbonyl oxygen and the amide nitrogen, they are trans to each other. You can see this oxygen is arranged above and this hydrogen is arranged below. So this opposite arrangement is referred to as a trans. Okay, trans configuration. So the carbonyl oxygen and the amide nitrogen, they are trans to each other, and this arrangement or this configuration is actually favored energetically. Okay, it is favored energetically because it results in less steric hindrances between the non-bonded atoms in the neighboring amino group. So there is less steric hindrance. That is why this configuration is more favorable. That is the trans configuration of the carbonyl oxygen and the amide nitrogen. Now the angle, okay, the angle which is formed between the alpha carbon atom and the, you can see this one, the alpha carbon atom and the nitrogen, okay, this bond here or this angle here which is formed between these two is referred to as the phi angle, and phi is denoted by the symbol. Okay, let me show you this one. Okay, this angle which is formed here is denoted by phi. Okay, so this is phi angle, and the bond which is formed between this alpha carbon atom and this carbon atom here is denoted by psi. Okay, so we have, sorry, phi and psi. Now in 1930 and in the year 1930 and 1940, okay, there are two scientists, that is Linus Pauling and also Robert Corey. Okay, so these two people, they determine the X-ray structure of several protein molecules, because we know that proteins, they are made up of amino acids which are joined together in this way. So these two scientists, they determine the X-ray structure of several proteins and they propose the essential features of a peptide unit, okay, of a peptide bond. Now here, since the peptide is formed between, you see here, this is the first amino acid, so a bond is formed between this carbonyl oxygen and the amide nitrogen of the second amino acid. Okay, so this is the first amino acid, this is the second. So you know that the peptide bond is formed always between this carbonyl oxygen and the amide nitrogen, this one. Okay, so this peptide group is a rigid and planar. Okay, it is rigid and planar as a result of resonance. What they have found out that this bond here, it actually acts as a partial double bond. Okay, it acts as a partial double bond, that's why it is rigid. And this peptide bond, this peptide bond here, it has about 40% double bond character. So it acts as a double bond, a partial double bond, and therefore the rotation about this bond is not possible. Okay, the rotation about this bond is not possible, because when we study the structure of proteins, girls, you will see that they have different kinds of structures. We have the alpha helix, okay, then you have the beta sheets, okay, I'm just giving example, the alpha helix, the beta sheets, then the beta bends which will connect these two sheets, okay, parallel or anti-parallel sheets. We have beta sheets which are arranged either parallel to each other or anti-parallel like this one, and there will be another structure term as beta bends which will connect these sheets. So we have many structures of proteins which you will see they are arranged in these ways. So you can see that, suppose here for alpha helix, okay, the arrangement, when the amino acids they are joined together, they have to fold, right? They have to fold in this configuration. So that means there should be a rotation. They cannot be straight like this, okay, straight and planar like this. That means there must be some place here where these bonds which are present here, they must rotate in order to bend like this, okay, in order to achieve this configuration, or in order to achieve this configuration or this beta bend configuration. How can they bend if they are rigid or planar like this? So there must be a place where they can rotate and bend so that they will form these structures. When you study the protein structure, you will see these. So just remember for now that this bond, it cannot rotate, okay, it cannot bend or it cannot rotate at any angle. Why? Because it is rigid due to because this bond, it acts as a double bond, okay, it has 40% double bond character. So since it acts as a double bond, it is rigid, and it therefore rotation around this, it is not possible. Again, same thing here, this amino acid is joined to this third amino acid through this bond, right? So again, this bond and this bond, that means they cannot rotate. Rotation around these bonds is not possible. Same thing when another amino acid will be joined here. So same thing. Now this leads to only two rotational degrees of freedom. That means only these bonds here, they can rotate. There are only two possible ways, this one and this one, that is the phi and the psi, okay, because they are single bond. Even here, just remember, you see that it is a single bond, but just remember that these bonds here which join one amino acid with a two amino acid, that bond, it always has 40% double bond character, so rotation around these is not possible. Instead, the amino acid, it can rotate only at this angle, phi and psi, okay, even here. So rotation is possible only at these bonds because they are single bond. So I hope that is clear, okay? Because now the X-ray diffraction studies of the peptides by these two people, by these two persons, it has shown that the peptide CN bond, okay, this peptide CN bond here, it is somewhat shorter, okay, it is somewhat shorter, around 1.32 angstrom. This is the measure of the this bond here. It is somewhat shorter than this normal CN bond. You can see here also we have carbon nitrogen bond, Okay, so this normal CN bond is actually 1.47 angstrom. But when one amino acid is connected to the second amino acid through this bond here, this bond is somewhat shorter than this normal CN bond. In simple amines, the atoms associated with the peptide bonds, they are coplanar. Okay, why? Because it indicates a resonance of partial sharing of two pairs of electrons between this carbonyl oxygen and the amide nitrogen. So there is a partial sharing of two electrons, of two pairs of electrons, okay, between these two. So as a result, the oxygen now will have a partial negative charge, and this nitrogen, since it loses these electrons from here, it will have a partial positive charge, because it's not that it loses the electrons, it share the electrons, okay, these electrons it share with the oxygen. So there is a sharing of a pair of electrons between these two. So that is why this, since it gets more electron due to nitrogen, it will become partially negative, and this will become now partially positive because proton is more. Okay, it has shared its electron with the oxygen, so it will have a partial positive charge. Now, since positive and negative are present, that means here since these two, they has a partial negative and nitrogen has a partial positive charge, setting up a small electric dipole. Okay, it will act as a small electric dipole. So you see here, okay, I will just draw this part only. This is the first amino acid, the carbonyl oxygen of the first amino acid. It form a bond with the amino group of the second amino acid. So since this nitrogen, it has these electrons, it will share these electrons. It will not give away, okay, to form the double bond here. No, just remember, it shares these two pairs of electron. So here it will become just partially positive and this will become partially negative. Okay, so acting as the small electric dipole. Now this is referred to as the resonance, okay, in a peptide bond. Now the 6 atoms of the peptide group, they lie in a single plane. Okay, they lie in a single plane with the oxygen atom of the carbonyl group and the hydrogen atom on the amide nitrogen, they are trans to each other. This I had already mentioned here. You can see all these groups, all these 6 atoms, okay, nitrogen, carbon, hydrogen, and this side chain, this oxygen, this carbon, all these atoms, they are arranged in a single plane. They lie in a single plane, but with the oxygen atom above and the hydrogen atom below. The oxygen atom of the carboxyl group and the hydrogen atom of the amide group, they are trans to each other. Okay, when they are opposite to each other, that is referred to as trans configuration. Okay, now from these two findings, okay, about this bond here, which cannot be rotated. Why? Because it acts as a partial double bond character due to sharing of electrons, and also these bonds here, the phi and the chi bonds, they can be rotated. So from these two findings, Pauling and Corey, they concluded that the CN bond, okay, that means this bond here, it is unable to rotate freely. Why? Because of their partial double bond character. That means rotation, it is permitted or it is allowed only through these. Okay, why? Because these, they are single bond. Alright, so in an amino acid, because we know that amino acid, the structure of amino acid is like this, right? C double bond O, and this will be joined to the next amino acid, C double bond O, O, H. So suppose this is a dipeptide. These two amino acids here are bonded together by this peptide bond. So since this, it acts as a partial double bond so it will not be able to rotate. That means rotation is only in this one and this one is possible. That is within the amino acid itself. And these bonds, these angle here, they are referred to as the Psi. The CN bond is referred to as the Psi, and the CC bond is referred to as the Phi angle. Okay girls, so I hope it's clear till this part. Okay, so besides these two persons, there is another person named G.N. Ramchandran. He studied these bond angles, okay, in amino acids and also this peptide bond. And he gave a plot, okay, he gave a plot, a presentation of these bond angles. Okay, he studied these bond angles involving the dipeptides, and he presented, based on the rotation of these angles, he presented a plot, okay, and that plot is therefore referred to as a Ramchandran plot. Okay, and this plot, it is based on many assumptions given by him. So let us take a look at the plot given by this Ramchandran. Okay, now you can see here, this is the plot given by him. Okay, so his assumption was that when he studied these bonds, there are many assumptions given by him, such as the peptide bonds. These peptide bonds, they behave or they act as double bond, okay, and the rotation is possible around only these two, that is the C alpha and nitrogen, and also C alpha and carbon. So these two bonds, that is the Psi and the Phi angle, the rotation is possible only between these two because they are single bond, while rotation is not possible around these two because they act as double bond. And the range of rotation in these two, the Psi and the Psi, is between minus 180 degree to plus 180 degree. So he gave this plot based on that assumption. Okay, and also by convention of the formation, okay, corresponding to this angle here, the Psi and the Psi and the Phi angle equal to 0 degree only when the two peptides, they are in the same plane. Okay, they lie in the same place, as in this case. They lie in the same plane and they are connected to a common alpha carbon. And here he gave this plot. You can see this plot here, actually you can see that the shaded areas, okay, these shaded areas here, they represent the combination of Phi and Psi angles, okay, which are more favored, okay, sterically favored. So here, as you can see, this shaded region here, it favors the beta sheet conformation. Okay, that means the proteins here, they favored the beta sheet conformation. And here you can see right-handed alpha helix. Here in this plot, any of the configuration, whether it is beta sheet or alpha helix, it is not favored. Okay, and here you can see this plot here, it represents the shaded region. Here it represents that the alpha helix, which is left-handed, because when you study the structure of amino acids, you will see there the alpha helix is right-handed. Okay, but then left-handed also is present, but it is rare. So whereas these dark shaded region here, they represent the most favored combination. That is, you can see here minus 60 degree, and from here also around minus 60 degree. So he gave this plot to see the possible configurations of amino acids in the proteins. Okay, so all these possible combinations of rotation of this Phi and Psi angle, this plot is known as a Ramachandran plot. And from this plot, what we can conclude it that, suppose if the Phi and the Psi angle, they are at 0, okay, they are at 0, then that one is not allowed sterically. Okay, it is not favorable for any configuration if it's at 0. Both the Phi and the Psi angle, if it's at 0. This one, it represents the Psi and this represents the Phi angle. The y-axis, remember, it represents the Psi, and this one, it represents the Psi angle. Okay, the x-axis. So here, if both the x and the y-axis is 0, then that one, that configuration is not allowed. Okay, that means that rotation is not allowed where both is 0. Now suppose if this is minus 60 degree and this Psi also is, this is plus 60 and this is minus 60, then it will produce the left-handed alpha helix. Okay, you can see here, this is plus 60. Okay, this is 60 plus 60, whereas this one is also plus 60. So when both are plus 60 degree angle, they rotate at plus 60 degree, then this left-handed alpha helix will be produced. Okay, it will be produced. And suppose here again, for example, if this angle, this Psi angle is minus 60 degree to 100, okay, if it is minus 60 degree to 100, and also this one, also this Psi angle is minus 60 degree, they will produce the right-handed alpha helix, okay, which falls under this quadrant. So in this way you can see, for example here, if this one is minus 90 degree, okay, around here to 180, from minus 90 to 180, and this Psi angle is from 90 to 180, then this configuration is favored. Okay, minus 90 around, 90 degree is around here. You can see this dark here, 90 to 180 till here. You can see this shaded region in dark, okay, dark shaded areas. Then beta sheets will be favored. So it depends on the rotation of these two angles. They will form different structures of proteins, okay, based on this Ramachandran plot, whether the right-handed alpha helix or the left-handed alpha helix or the beta sheets will form. Okay, so based on this plot, and this plot is referred to as the Ramachandran